For centuries, artifacts like the Shroud of Turin existed strictly on one side of a thick historical wall. They were revered objects of theology, heavily guarded and kept entirely separate from empirical science. Handing ancient relics to modern, secular forensic laboratories should logically result in one of two things: the discovery of a medieval hoax, like paint or animal blood, or the identification of a standard, verifiable human biological profile. But that barrier between faith and science was breached when researchers began securing permission to run independent laboratory tests on samples taken from multiple different miraculous events. The independent labs did not expose a series of historical hoaxes. Instead, plunging into the cellular tissue of these artifacts revealed a recurring biological anomaly, a signature that did not fit expected patterns of typical human genetics. Subjecting these sacred objects to the strict rigors of the scientific method hasn't debunked them. It has opened up a forensic mystery that demands a closer look at the data. To understand the scope of this anomaly, we have to look at the investigation's methodology. Researchers treated these miracles strictly as raw, isolated datasets, scanning them purely for statistical overlaps. The earliest data points come from old Italian case files. We're looking at ancient Eucharistic miracles, specifically events recorded in Lanciano and Bolsena. Scholars like Father Spitzer, working with the family of Blessed Carlo Aquitas, began compiling these historically isolated events into a single comparative study. Then they added modern data points to the mix, the relatively recent Eucharistic miracles that occurred in Buenos Aires, Argentina, and Tixla, Mexico. The artifacts from Italy and Latin America are separated by oceans and centuries of history. Physical cross-contamination between these samples is an impossibility. By applying blind forensic testing to objects separated by centuries and continents, the researchers discovered a recurring biological pattern that defies the odds of coincidence. The first layer of empirical testing applied was modern hematology. Technicians placed the tissue under the microscope to run a standard blood typing analysis. When the results came back, every single scientifically tested Eucharistic miracle where blood could be determined yielded the exact same result. The blood from the ancient Italian relics and the modern Latin American events all tested as type AB. This identical result matches the blood type found on the Shroud of Turin. This diagram outlines the ABO blood group system. Notice type AB sits in the center. In medicine, individuals with type AB function biologically as the universal recipient, safely receiving blood from any other type without immune rejection. This blood type takes everything in. It is a striking biological parallel to the theological concept of Christ receiving all of humanity into himself. Beyond simple blood typing, the presence of red blood cells and tissue means that modern laboratories should be able to sequence a complete DNA profile. Genetic sequencing, however, presents a different result. While the tissue registers as living and human, the standard process of extracting a complete sequence fails to return a standard result. Look at this standard human karyotype chart. A healthy genome requires 46 chromosomes, organized into 22 distinct pairs. Yet when technicians mapped the relic samples, they found only 22 single autosomes total, plus a lone Y chromosome. This creates a biological paradox. It registers as human and living, yet missing fundamental genetic building blocks. This incomplete genetic code is not the result of a single lab error. It is a reproducible, unified biological profile found across independent relics scattered globally. For experts like Father Spitzer, this extensive cross-examination provides a tangible, measurable physical parameter for phenomena that have historically confined to theology. When modern medicine examined these sacred artifacts, it didn't find the standard human biology expected. It uncovered a biological signature that defies the established rules of genetics. The physical reality of the samples presents a unique challenge to biology. Tissue that registers as living and human, despite an incomplete sequence that should, by any standard measure, prevent its existence. By testing these relics under secular science, we have moved past theological speculation and into a measurable, recurring biological profile that currently sits outside our understanding of human genetics.